The human body is remarkably resilient, but it has hard limits. When complex tissues like the shock-absorbing cartilage in your knees or the delicate valves in your heart fail, they do not grow back. They are lost permanently. For decades, the primary solution has been to install cold, non-living machinery. Tissue engineering attempts to approach this problem differently, constructing living human tissue from scratch to replace those failing parts. You have likely seen the headlines hyping the imminent arrival of 3D printed organs, yet hundreds of thousands of people remain stuck on massive, unmet transplant wait lists worldwide. The delay keeping lab-grown organs out of hospitals isn't a lack of biological raw materials. We have the cells. The true obstacle is an escalating battle against physics, geometry, and structural complexity. Tissue engineering is a systems problem. Success requires that cells, chemical signals, and physical environments all align to create a functioning, living whole. To understand the scale of this challenge, we have to look at the complexity gradient. We start at the lowest level, flat, two-dimensional tissues with simple geometries, like skin and the outer layer of the eye, the cornea. The raw materials for these flat tissues come in two forms. Somatic cells, fully mature cells from a patient, are safe but difficult to multiply in the lab. Alternatively, scientists use mesenchymal stem cells, or MSCs, which are highly flexible adult stem cells. MSCs serve as the foundational workhorses of this first level. They perform two distinct jobs. They can differentiate to form the actual physical tissue, and they actively secrete powerful localized chemical factors that reduce inflammation and promote healing. Because these tissues are structurally flat, growing them in a petri dish is already a routine clinical success. Provided with the right growth factors, these cells will actively divide and spread uniformly to form a continuous sheet. We can reliably engineer flat tissues today because their simple, two-dimensional geometry grants every single cell completely unrestricted access to life-sustaining oxygen and nutrients from the fluid surrounding it. The difficulty spikes at level 2. This involves intermediate tissues that require three-dimensional, hollow, or tubular architectures, such as blood vessels, tracheas, and bladders. Here we hit a geometric bottleneck. If you place a cluster of cells into a nutrient bath without any intervention, they will simply pool into disorganized flat sheets or clumps. Biology does not spontaneously form a hollow tube in a petri dish. This diagram illustrates the necessary solution, a scaffold. By introducing a physical, porous 3D structure that mimics the body's natural extracellular matrix, researchers can actively dictate the shape the cells take as they attach and migrate. Builders have two primary material choices for these frameworks. The first is natural, using donor organs washed of their original cells, leaving an organic matrix. The second is artificial, synthetic hydrogels and engineered polymer meshes. Having the right structure is only half the battle. Before they are seated onto a scaffold, stem cells undergo directed differentiation, where they are exposed to a strict sequential cocktail of chemical signals to mature them into the specific cell type required for that tissue. Elevating a culture from a flat puddle of cells to a functional three-dimensional tube requires both rigid architectural blueprints and precise chemical sequencing. The highest tier of biological complexity involves thick, solid organs packed with intricate internal microarchitecture. Take the solid liver, the complex kidney, or the human heart. Looking at this cross-section of a solid organ shape reveals the core limitation. Passive oxygen diffusion only penetrates a few millimeters deep. The cells on the outer edges breathe fine, but the deep interior cells quickly suffocate and die. The technological countermeasure to this restriction is a perfusion bioreactor. These mechanical systems forcefully pump oxygen and liquid nutrients deep into the artificial vascular networks carved within the tissue. These bioreactors must also simulate the physical stress the organ will face inside the human body. By applying rhythmic mechanical loading, the machine forces the lab-grown tissue to strengthen and mature correctly. Constructing an entire solid organ requires an astronomical volume of raw material. To meet this demand, scientists use highly flexible pluripotent cells, like iPSCs, which possess the unique ability to multiply aggressively and turn into any cell type in the body. That extreme flexibility carries severe biological risk. If even a tiny fraction of these cells remain undifferentiated when implanted, their aggressive growth rate causes them to rapidly mutate into dangerous tumors, known as teratomas. Solid organs remain the most difficult tissues to engineer, requiring the flawless synchronization of millions of highly volatile cells, sustained entirely by artificial life support networks. The biological ingredients required to build human organs are well documented. But scaling those ingredients from a controlled Petri dish into the chaotic environment of a living human body is an immensely perilous tightrope walk. Before clinical deployment is even an option, researchers must pass severe safety checks. They must guarantee absolute zero tumor formation and ensure that any artificial biomaterials integrate seamlessly into the host without triggering an immune rejection. The complexity gradient dictates our current reality. Two-dimensional skin sheets are actively saving lives in burn wards today, while the intricate, plumbing-heavy architecture of vascularized solid organs keeps them locked in the laboratory. Yet the trajectory of the field is clear. Medical science is moving past the era of simply cutting out disease or implanting metal mechanisms to compensate for failing parts. Regenerative medicine is the audacious attempt to hijack the human body's own developmental logic, rebuilding life from the ground up, one material.
If you like this video, give us a thumbs up